Hello, everybody. Welcome to today's CIC lecture given by Dr. Sophie Vock. So Sophie Vock was born on a very stormy night in Amsterdam, the Netherlands. She majored in artificial intelligence and social political philosophy at the University of Amsterdam and finished MSc in cognitive and brain sciences at the same university. Following, she pursued a PhD in social neuroscience at the Max Planck Institute of Human Cognitive and Brain Sciences in Leipzig, Germany, where she investigated the structural basis of the social brain and found that cortical thickness changes as a function of social mental training. She was awarded the Otto Hahn Medal and Award for this work. Following, she was a postdoc with Professor Simon Eickhoff in Forschungszentrum Jülich Heinrich Heine University, Düsseldorf, Germany, where she worked in imaging genetics. Here, she found that multiple heritable axes organize the human macaque cortex and macaque cortex. Currently, she's a group leader of the cognitive neurogenetics group at the Max Planck Institute of Human Cognitive and Brain Sciences and at the INM7 in Forschungszentrum Research Center Jülich. Her hobbies include running and building Duplo trains with her kids.
So thanks, Nadia, for this introduction. I'm Sophie, and in the next 45 minutes to an hour, I will tell you a little bit about the work that I've done or am doing, and that also now my team is happily doing, or maybe they're not happy, but I really love what they're doing. So yeah, I hope you like it. And yeah, as discussed before, please ask questions maybe at the pause moments after every capital, so to say. Because that's maybe just easier. So I have summaries after each major point. So one of the questions that keeps me occupied is what are other people thinking? But to give it a more scientific meaning, I also wonder how are people thinking and what is the stuff that shapes our thought? And for this, I look at brain structure and how the anatomy and also the whole building, the architecture of the brain also creates a way that we can think in sort of a certain way. So we have certain functions allocated roughly to different regions. So that's sort of the framework. And this is a photo of my friend from kindergarten, who's now a photographer, and I really like her work, but that's maybe a side note. So the brain, the cortex in particular, so yeah, apologies for subcortical fans, I will mainly talk about cortical results here, has various structural zones with a different laminar profile. So you have the paralimbic areas that are here in green, that have more agranular profiles, and then you have higher order association regions, modality-specific unimodal association regions in yellow, and then in blue, more ideotypic primary areas that have the clearest granular structure. And this difference in structure may also a way to understand differences in processing that is underlying it. So that's one part of my thinking, I guess. So Mesulam in 1998, but also other people have postulated that this creates a certain hierarchy or heterarchy, so a sort of network-like processing way to come to this abstract thought. And indeed, using, for example, a gradient approach, looking at these primary axes and dimensions in the brain, you can also understand how different levels of abstraction and also different levels of microstructural abstraction may relate to this abstract, from perceptual action thoughts to more abstract thoughts that are not directed to the immediate environment. And I guess the work of Margulis et al. In PNAS was in that sense rather important because it proved also a method that captures this more theoretical framework in an elegant way. Another paper or approach that I really like here is this broader idea that the human brain is one of a kind in some ways, but in other ways, not really, it's because all animals have brains, and it helps us also to understand how humans can think by comparing our brains to the brains of other primates. So for example, here you see also these important commonalities here. So you have all these similar types of primary regions, whereas what is between may vary. So this is another thought that I find really interesting, and I'm also now pursuing. So the first part will go a little bit about how a brain is built and speak about my work there. So for this, I use cortical thickness, which is like, to put it simply, you have a T1 MRI and you compute the distance between the vertices at the white matter surface and the tile surface. I don't know, Nadia, do you see my mouse or not? No? Okay, then I'll just use my words.
Sorry, yes, we do. Sorry, I can see.
Oh, you do, okay. Okay, good, but it doesn't matter so much, but at least you have like then a thickness of the cortex. And what is important, you have different methods to do so. So I guess that you guys in Montreal like CVIT, other people work with FreeSurfer or with CAD, and all these approaches, they give more or less the same estimations, but not quite the same. And especially in these areas that have the light here in black, there are like some challenges respective of the method. So there are some challenges with the method, but in principle, they can be compared, and the rough estimate is similar. So for this study, I looked at cortical thickness covariance profiles in humans and in macaques. And yeah, structure covariance was there like an important metric, because it's a cheap way, I would say, maybe to get at like this idea of what is like the relationship between the anatomy of two regions. So the concept is that if you have a seed region, for example, here in the anterior insula, and you covariate with another region for a whole group, you see that it can have a positive correlation, meaning that when region A gets thicker in person A, it will also get thicker in person A in the other region, whereas another person may have a relatively thin region A and also a relatively thin region B. You can see this group level covariance. So this has been observed already in, if I'm correct, 2006. And the question was a bit like, what could it mean? So Schmidt in 2008 could show that it already has like also this genetic correlation. So those regions that go very across a group of individuals are also regions that have maybe a shared genetic basis underlying it. But it also relates to maturation effect, which doesn't, exclude that it is genetic, but you also see that regions that have a covariance profile, they also mature in a similar way. So my idea was to investigate these large-scale axes underlying structural covariance using this gradient approach, which simply you have a matrix, in this case, a covariance matrix of thickness covariance across individuals in the human connectome project data. You normalize this matrix and then you order it along a gradient to look at the principal and the secondary axis that make these connectivity profiles similar. So in this ordering, regions get close to this space to each other if they have a similar connectivity profile, and more distant when their connectivity profiles differ. So here, my main interest was also to investigate these genetic axes. So for this study, I used also the twin setup of the Human Connectome Project. And these are actually, there are monozygotic twins. And by comparing twins with each other, you can compare monozygotic and dizygotic twins to unrelated individuals. And if something is genetically correlated, one can infer that those people that have more genetic similarity would also show higher similarity in their covariance profiles versus those that have less genetic similarity with each other. So indeed, we could show that structural covariance has a stronger correlation with the genetic correlation and a weaker correlation with environmental correlation, meaning that the covariance between regions is driven by genetic factors to a large extent. Then I used this genetic correlation matrix and this gradient approach to infer what are the main axes that shape this genetic correlation. So regions that are at the same level of this axis, they show a high genetic correlation and a low genetic correlation with regions at the other end of the axis, as you also see here in this plot. So regions at the same level, they show like a maximum of genetic correlation, whereas the more distant they get to another region, the less genetic correlation it has with a certain region. And what I found in this data set and also in others was that there are two main axes. One goes more from unimotor regions to especially anterior frontal regions, and the other one goes from more superior regions to here more paralimbic and also inferior regions, as you see here in the temporal lobe and the insula. Then I went one step further to also investigate, like I show these axes are related to genetic correlation, but if this is also something that is phylogenetically conserved. And here I worked also with Ting Su, who was an important co-author in a paper looking at macaques. So here I did the same approach. So I looked at thickness covariance in macaques, and I again looked at these main gradients. And what I found was, again, two major gradients, one going more from here, superior regions again to inferior temporal regions, and the other one going from like motor regions, sensory regions to frontal cortex, as you see here. And I colored them like in a way that makes you think, okay, this one region, like, is this pattern then related to the human pattern? But you can also formally, like, this was mainly also driven by like looking at the patterns, and you see, okay, here's this clear frontal point, and here's also like this clear. So that's why I interpreted like this, but I wanted to also formally test this relationship. And for this, I used the metric of a joint alignment, which Ting also published in her 2020 paper. And it's this joint embedding, and this is like a quick visualization of how this would go. So the idea is that you create similar anchor points between humans and macaques, and with this you can align these matrices to each other, and then also map back the homologs between species. Along the different dimensions too. So this would be the second dimension. So we'll not go through all the dimensions, but you get maybe the gist of the idea, and I can also recommend reading the amazing paper that she wrote. So I transformed the human gradient to macaque space to compare both gradients with each other. So this is then how it looks, and you see it looks a little bit different because macaque brains are different. And then I found that indeed the human principal gradient that was posterior to anterior showed a positive correlation with the first gradient, but no correlation with the second gradient in humans. The second gradient in macaques and the first gradient in humans showed a positive correlation, whereas the second gradient in humans did not correlate with the second gradient in macaques, whereas the secondary gradient from superior to inferior regions in humans was positively associated with the first gradient in macaques, but not the second. But what does this mean? Because yeah, I show patterns, great. So then like there has been a lot of work in what organizes the cortex and what are the differences. So for example, the difference between posterior cortex and anterior cortex, they relate to neuron density, with the exception of primary regions, as you see here, along this principal axis. So the neurons per millimeter per 3, but it also relates to neurogenesis and time. So there's a difference between posterior and anterior regions in various features, so to say. So neurogenesis ends later in posterior regions relative to anterior cortex, but there's also smaller soma, shorter processes versus larger soma, longer processes. So there's all these kinds of differences between posterior and anterior, which may be a reason why there's a more genetic correlation between posterior regions with other posterior regions versus anterior. At the same time, we also show that this anterior to posterior pattern relates to the principal gradient. So it has some relationship with unimodal versus transmodal dissociations, which has again been found to be something that has changed during primate evolution when comparing functional organization in macaque to humans, but also when looking at surface-based expansion in chimpanzees. So this could be like one interpretation of this posterior to anterior pattern that I found. However, the inferior to superior pattern cannot yet be explained by that. And for this, we also had to think about the dual origin model, which postulates that there are two origins of the cortex, so to say. One is the archicortex and the other one is the paleocortex. And from these agranular origins, this granular specification emerges, so in a wave-like fashion, so along these arrows, so to say. And indeed, I made then a model of the dual origin by probing the distance for both origins So here is the paleocortex, here's the archicortex, and here I tried to also go along the arrows. And I found indeed that genetic correlation is strongest along these dual trends, and that even more so the distance of each origin related positively and negatively to the second trend, but not the first trend. Taking two models into account, one would be like taking it as a binary account, and the other one would be like more a gradual transition. And the same held true for the first organizational pattern in macaques, where you also see this interaction between distance from paleo and archicortex. So yeah, these two patterns that I observe in genetic correlation between regions, like on the one hand relate maybe to functional hierarchy and anterior posterior patterning, but at the same time, the second pattern relates to the dual origin. So are there questions now, or shall I just continue?
I don't see any in the chat. If anybody wants to raise their hand, otherwise I had a couple questions. I was wondering about, first of all, the joint embedding technique to compare brain maps. Is it like, I guess, I mean, it's obviously very different from like the Alexander Block spin test. Is the point of the joint embedding technique to circumvent the problem of comparing two brain maps with different number of vertices? Like, could you just do a spin test between gradient 1 in the human and gradient 1 in the macaque? Or, yeah, I guess I was wondering about like what the...
Yes and no, but you wouldn't know if like the same vertex corresponds to the other vertex in them. Like you need some way to bridge human to macaque space, right?
Yeah.
So this is what you do with this joint embedding, where you looked at shared anchors and then you embed one brain in the other space, preferably human to macaque, under the assumption that macaques have things, or humans have things that macaques don't have, so it's easier to go in that direction.
Then I get Molar.
Well, let's go with Trezana's question first.
So Trezana was asking, just wondering what your definition of archicortex and paleocortex is.
Yeah, very good point. So in this paper, I looked also at the literature, and paleocortex was the piriform cortex as defined in the Glasser Atlas. And for archicortex, I actually took regions along the archicortical trend, stretching from the hippocampus to posterior cingulate areas, and then created the distance, because this map would then most closely follow the patterns that was observed in the literature, I believe also in Panja 2015. But at the moment, I'm also working on a project to better define this dual origin axis, not in this naive way and distance based, but more using cytoarchitectural approaches.
So my question is somewhat related to Nadia's. Would that technique work well if you were trying to compare, I mean, not just the cross-species homology piece, but also maps that come from individuals where topologies may be questionable? So if you think, for example, an infant brain versus a adult brain, for example, or as being on two ends of that spectrum as well, would that be another place where you'd want to use something like that? Because certainly the spin test there doesn't work quite as well.
Yeah, I guess I would say yes. I think that would be helpful to quantify like the different spaces that like a brain can be in. And I guess it also relates to the question of normalization, right? Can you map everybody to the same template, or do you use like an infant specific template versus adult template, but then how to compare? So yeah, I guess this is one way to potentially bridge these different template spaces, although, like as every method and approach, like I like it a lot and I use it a lot, but there's also other ways that try to bridge it like into this broader common space idea.
Olani Kupo was asking, the dual origin hypothesis, is that related to how the brain develops over the lifespan, or how it develops through phylogeny evolution?
It can be applied to everything and anything. So in principle, it's more an evolutionary theory, but in some ways, ontogeny mirrors phylogeny. So one would expect that it also would relate in some way to development. And I will also at some point, I think in one of the later slides, I also come back to this idea again. So maybe that helps. But yeah, it is. There's also a whole book of Panja of 2015 on the dual origin. If you're interested, I can recommend reading it. It's rather nice and interesting.
Thank you. I think that's it for questions for now.
Okay, so then I think enough people could watch this GIF and wondered, what is this about? So I guess, the idea is like, what are at least these organizational features that relate to human cognition? And one of the other patterns that has been like discussed in the literature is asymmetry. So like it's rather like classic literature, like take this Geschwind paper from 1968, have shown that there are differences between left and right hemisphere, and that they may relate also to our ability for language, which is, language is a unique human skill, and like animals can communicate and in awesome ways too, but like this talk that I'm giving now could be by no means be done by an animal. So that's why I was also interested in asymmetry, and Bin Wan, who works in my lab, did an amazing job in a paper that will be in preprint this October on the differences in organization between left and right hemispheres, using again a gradient approach and using functional connectivity in the human connectome sample. So what it did is that we looked at the difference between individual functional connectivity patterns of left to left connectivity versus right to right connectivity versus left to right and right to left connectivity, to compare these inter and intra-hemispheric connectivity patterns. And to do so, we again used a gradient approach to see if these main axes of organization were differing between left and right hemisphere. So it's a data-driven approach to see what is different, and overall you see that there's a high correspondence between left and right patterns. So at first glance you would say, okay, asymmetry is like more like the cherry on the cake, than that it's a main principle, because overall left and right are rather symmetrical. But to me, or like I hope this project will show you that the cherry really makes a difference, because if you compare intra-hemisphere differences, you see that some regions have a preference for the left hemisphere and are higher up this hierarchy from unimodal to transmodal regions, meaning they're closer to the default mode region. And these are the blue regions, as you see here. And interestingly, they also overlap with regions that are known to be involved in language. Whereas other regions, they show a preference to be organized towards the right like hemisphere hierarchy. So they're higher up in the rightward hierarchy. And this pattern was mirrored to some extent in the interhemispheric patterns, but there are some differences between intra-hemisphere versus inter-hemisphere connectivity. But for both patterns, we see that this is highly significant looking at individuals. So you see here, all these red maps indicated that there's a much higher leftward asymmetry across individuals, except for the visual region. And here you see it again summarized as a function of networks. And here you clearly see that the language network is most leftward organized, whereas the frontal parietal network is most rightward organized. Then we also looked at heritability. And what is interesting is that this measure is rather heritable. So not all regions are heritable. And here you see like the FDR corrected patterns. But you see that various language regions, but also sensory motor and like frontal regions, and even more the interhemispheric connections, they are largely heritable, meaning that these individual differences that you see in asymmetry of functional organization are in some part governed by genetic factors. And though there is not a direct link, I believe that the interhemisphere shows some relationship between heritability and asymmetry. It is not that consistent. And what is interesting is that you see that some language regions, which are here colored in greenish, they are highly heritable. So you see clearly they are really outliers, but other language regions are really asymmetric, but not so heritable. And this is also happening along a posterior to anterior gradient, whereas those regions that are more posterior are more heritable and asymmetric, whereas those that are more anterior are less heritable, but still asymmetric. And you see here in yellow, it's not super clear, but you also have the same pattern for the right hemisphere. So then we also wanted to know if this pattern was phylogenetically conserved. And for this, we used the same approach in the macaque data, where we have here the intra and interhemispheric patterns. And then we compared the macaque data to the human data aligned to macaque space, as you see here. And what is nice is that you see both patterns show a quite nice correlation, like 0.39 like is highly significant, and it seems that this asymmetry is somewhat conserved across primate species, which is also in line with other research showing that there is some form of asymmetry that is shared across primates. On the other hand, like trying to quantify difference between humans and macaques, and these are like subtle differences, and like overall it's nice that we at least see this conservation. But what I also like at the same time is that you see that language regions are way more leftward asymmetric in humans relative to macaques. So you see other networks, they are like roughly similar, although somatomotor is way more asymmetric in macaques relative to humans. And to me, this is an intriguing finding to see that this functional organization asymmetry is so much more pronounced in humans relative to macaques. So yeah, to sum up this work is that asymmetry seems to be a key feature of the primate brain, and in particular language network are leftward asymmetric in humans, but the control network show rightward asymmetry. And this functional asymmetry is heritable, and we could also replicate this in the UK Biobank sample, whereas interestingly, we show that this leftward asymmetry is more stable maybe because this rightward asymmetry is not present in UK Biobank, or at least not that strong, which may also relate to the fact that this is an older population. And there are also some ideas about how asymmetry of organization may change during the lifespan. So are there any questions about this? Or shall I just continue?
If there are no questions, I had a question. I was wondering, amongst your human participants, would you have information on how many languages they speak, read, or write? And do you think that that could be an interesting to see, like, whether somehow the number of languages you know affects that asymmetry?
I don't believe that this is a like, I know that other studies have shown that this asymmetry may relate also to some semantic processing, individual differences. I don't know, in the Human Connectome project, there's not such a code for language ability. It may be that people with more pronounced asymmetry have like different language properties, but here we also mainly looked at the mean maps and looked at the mean differences. But you're right that like obviously it also relates to individual variation, and yeah, so possibly people, the more language you speak, the more heavy your left side gets.
Molar?
Yep. To that end, I have a quick question. Are you going to, or have you thought about examining these properties in groups with neuropsychiatric disorders? So for example, there's pronounced differences in asymmetry that have been observed in ASD and schizophrenia. Do you know if that maps on to the type of metrics you're using here?
At the moment, the computer is running, calculating exactly this for an OHBM abstract.
Perfect timing. There you go.
So yeah, this is part of Bin's PhD work. So yeah, we are also exploring now how this relates to neuropsychiatric disorders and development. And yeah, there are some theories also saying that individuals with ASD, that they have less asymmetry, so they have more symmetric brains, if I'm correct. But yeah, I think asymmetry is a super interesting feature, and it also is associated with like efficiency of processing, and like, yeah, it's a fascinating phenomena, I think, and also like tricky at some levels to compare left and right. And here we use the Glasser Atlas because they mapped all the regions and the homologs in a nice way. But yeah, there are also obviously different approaches to this.
Yeah, thank you.
I had a question. This is maybe more, would be more better catered to zoologists, but I was wondering if you knew much about like how macaques communicate, like, because they still kind of communicate with some kind of sounds, right? I would guess, but I don't know.
Yeah, I think though that communication with sounds alone, it doesn't make a sentence, right? So I think this whole language networks in humans is also like, there's pretty nice work of Nicole Eichert and eLife, also comparing the arcuate fasciculus between macaques, chimps and humans. And there you see that in humans, this is most like most expanded. But yeah, it's I know that elephants also communicate like with like rhythms. So yeah, there's many ways of communication. So I would not like put too much weight on the specific functional meaning because it's only like indirect in some level. But what it is to some extent interesting that this asymmetry maps so nicely to the language network using data-driven approaches. But I think more is to be understood what it then means and yeah, how to perceive this. So the next topic will be about the hippocampus. So it's pretty hard to find a suitable GIF for the hippocampus, but the hippocampus is another cortical region that is super interested and also related to like shaping the organization of the cortex and also shaping its function and like also associated with memory related processing, but also spatial cognition and all these other interesting features that we humans have, right? So there has been already beautiful paintings of the hippocampus, as you see it here, nicely embedded by Fink de Azia in 1786. And the hippocampus is cool because here you see the development and it looks a bit like a worm, but it's like so infolded. And that means also that like understanding its anatomy and how it relates to like how the surface of the hippocampus also relates and like shows different features is also important to understand like what is its role in the brain and in relation to structure and function. So here you see it goes from the subiculum to the corpus ammonum to the dentate gyrus. So here you see it again. So what we do, we adopted a method previously developed also by Rainer Vosterwal and Pnas, looking at the different subregions of the hippocampus and then projecting functional data and microstructural data on the surface. So the first step was to validate or to see whether these maps would be heritable. And indeed, we show that like the functional connectivity analysis for the subfields, we show that these maps are heritable in a different, like there's some variations between subfields, but overall it's consistently heritable. And then we also looked at these gradients that shape the organization of these hippocampal subfields. So you see there are two gradients, one going from more posterior to anterior in each subfield, and the other one going more from inferior or from medial to lateral. So overall, these gradients themselves are not that heritable, which is maybe to be expected, right? Because you look at how these gradient values may vary across individuals, and whether this is meaningful or not. And this also relates a lot to the alignment of the gradients to see if the exact loading of these axes varies in a meaningful way. But at the same time, if you look at the heritability of the functional connectivity of the hippocampus to the cortex, you see that this is still organized along different gradients, in particular in the subiculum, and this is also the largest area, and this is maybe the most robust one in terms of TSNR. You see this difference between posterior and anterior regions, meaning that the posterior regions of the subiculum relate to different parts of the cortex or are heritable with different parts of the cortex than the anterior parts. So second, we then also looked at the microstructure in the different subfields, and we see that there's some relationship between the microstructure captured by T1, T2, and this second functional gradient, like that goes from lateral to medial, as you see here. And also microstructure is highly heritable. So you see here the p-values are all almost zero, meaning that the variation of this T1, T2 map along the hippocampal surfaces is also attributable to some extent to shared genetic factors across individuals. And this is also the same when controlling for a mean T1, T2 signal in the brain. Then we were interested at the relationship between the hippocampus and the isocortex. So for this, we created a covariance map of intensity using the T1, T2 imaging of the different vertices in the hippocampus to the cortex, creating this structural covariance map and thereby creating gradients, and also like using the method that I also used in a previous work, looking at genetic correlation and again creating gradients to see which regions genetically correlate with which other regions. So to start off with this covariance patterns is that we see again two gradients, one from posterior to anterior and the other from lateral to medial in all the subfields. And you see that these patterns also project to different parts of the isocortex. So you see that anterior regions, they have a higher covariance with particularly anterior cortical regions, whereas those that are more posterior relate more to posterior regions, such as the visual cortex. And the same pattern holds true for the CA and the DG, although this one is maybe least possibly also because it's the smallest and the TSNR is not the best. So there is some variation. It's hard to interpret if the variation is signal and noise to some extent. Then also having the same approach for genetic correlation, you see again that there's a differential genetic association between the posterior and anterior regions of the hippocampal subfields using our approach. And this extends also previous work using transcriptomic access, because here we show that at the individual level, there's also this differential genetic relationship between these subfields, and actually that it's very subtle, and that it means like that it was also helpful to look at the different subfields because the patterns are somewhat different between subfields and not all can be attributable to SNR factors alone. So then taking both structure and function together and by fusing the gradients, we aim to map like how does these general patterns that we observed and that are also partly shared between structure and function, how do they map on the cortex? And there we mapped this fused gradient with functional connectivity to see what regions of the isocortex are more related to the anterior or posterior part of the hippocampus. And there you see a rather nice distinction between like salience and frontoparietal networks versus sensory motor and DMN between the anterior and the posterior part. And what is also kind of intriguing in some level is that you see also a difference that whereas in the subiculum, it seems to be primarily dominant towards the blue regions, meaning that it has a high association with default mode, sensory motor, and also limbic regions. The dendrite gyrus, the GG, has a stronger association with dorsal attention, frontoparietal, but also visual regions, like showing maybe also a gradient among subfields that varies. And all these regions, they link to this third gradient in functional organization, which has also been related to task positive and task negative activity. So maybe in this way, different subfields of the hippocampus shape the organization of the brain in a slightly different way, but all link to this on and off task thought. So to summarize, functional and structural organization of hippocampal subfields is heritable, and there's a differential genetic association with the isocortex along these long axes, and functional networks associated with these axes vary as a function of subfield. And this is work of Shema Bayrak and will also be preprinted this month, I hope. So yeah, I'm very happy with what she has done. And also I find hippocampus is super intriguing and it's such a beautiful structure. So are there any questions? No. Okay, I just continue. Also, let me know if I said enough, right? So I can also stop at any point. So now, like going back a little bit more to function, and like function is tricky in the brain because resting state function in some way relates to function, but on the other way, it's also in some ways an anatomical measure. But here, I wanted to compare how the microarchitecture or cytoarchitecture of a specific region relates to its functional connectivity. So here you see that different regions have this different microstructural profile. And Casey Pacola, who was at the MNI and is now in Jülich, made this really nice metric that probes like this microstructural profile in the big brain, which is a histological sample, and also applied this then in 3T Tesla, showing that it is reproducible and that you can actually infer these microstructural profiles and also its similarity across the cortex. So regions, again, this is again a gradient with the same gradient value. They have a similar microstructural profile covariance, meaning that they have similar microstructural profiles. So here I correlated functional connectivity with microstructural profile covariance in the Human Connectome project sample. And I found that indeed there's a high correlation between most measures, meaning that regions with a similar microstructure are functionally connected in primary regions. But this is less so the case in other regions, such as unimodal, heteromodal, and paralimbic regions, which doesn't mean that structure and function are uncoupled per se, but they're differently coupled than in primary regions. And this may also relate to the fact that they have like a different relationship to heritable measures. So to infer to what extent these connectivity profiles of a specific parcel were under genetic control, I evaluated whether the mean pattern that also has been like used to probe a functional network is corresponding with the heritable pattern, indicating if a region has a high correlation with another region, whether it's also highly heritable or not. And you see again that this role, like this idea, is true in primary regions, but it is not true in especially heteromodal regions in both measures, and also correlates with this uncoupling, indicating that regions that have a different or uncoupling between microstructure and function are also regions that may be less under genetic control. And at the moment, I'm working on the revision, and so far I've been able to show that this does not really relate to TNSNR and is also stable with different repeated measure approaches. So I'm hopeful that this is not only because of noise, although noise is always a factor in our analysis, but also that it may have, at least at some level, a certain meaning of what is happening there. And then again, I compared this to macaque. So this is also, like, I guess, a major challenge in my current work, that I have only nice macaque data, but I really also need starting to look beyond this. What is nice that, again, I see that this correlation between microstructural profile covariance and functional connectivity is similar in macaques, yet also, like, you see that there's a correlation of 0.35, but there's also this dent here, meaning that these regions are highly correlated in macaques between structure and function, but not correlated in humans. And if you like look at this as a function of cytoarchitectural classes, but also functional networks, it is clear that this is in particularly true for heteromodal regions that involve also the default mode network, as you see here, and also the frontoparietal control network. So whereas limbic is also a little bit uncoupled in macaques, these regions associated with frontoparietal and default mode, they are more coupled in macaques relative to humans. So then this is sort of like, the paper got a bit long, so I tried to summarize this quickly. So I also looked at differences in the organizational axis between two measures. And what you see is that whereas functional organization goes from unimodal primary regions to the default mode network, this microstructure organization goes to more paralimbic regions. So there is somewhat a difference and a divergence between the main organizational axis of both measures. And this can also be seen in macaques. So you see here, like, these two mountains, you have on the one hand this heteromodal versus this paralimbic, meaning that these organizational axes can be diversified. And I then put together these two axes and projected task-based activations on it to see where they are placed in this 2D space of uncoupling, but also differential organization of microstructure and function. And what is interesting is that these regions, they are similarly uncoupled in humans and macaques, and they relate to emotion, affective, pain, phase processing, reward. Whereas regions that are uncoupled in humans but coupled in macaques, they relate to autobiographical memory, social cognition, episodic memory, which also, like, this is only based on task-based meta-analytic activation. So it's again group maps and group processes, but in humans, those regions that are uncoupled, they relate to these functions, and these are also regions that are least coupled with heritable measures. So it may relate to some way that flexible thought and the environment and also the cultural brain can interact with how we can think. And this also relates at some level, and I now realize I didn't include it, with the dual origin. So you see that this uncoupling relates to expansion of surface area, whereas this difference between functional organization and microstructure organization relates a little bit to the distance from archaea and paleocortex. So, like, to me, at least, this is interesting, but it is also like more of a speculation from my side that this may also be one of the reasons why you see these different patterns emerge in these different data types. So uncoupling between MPC and resting state functional connectivity in transmodal regions, this has a reduced relation with heritable profiles in heteromodal regions and a stronger coupling in heteromodal regions in macaques relative to humans. So this heteromodal region plays a central role here. And interestingly, these heteromodal areas are also associated with social cognition and episodic memory using these meta-analytical maps. So I guess time for some meditation, but also some questions.
I was wondering, are those heteromodal regions, they've probably been shown in neurodevelopmental studies that they expand a lot faster than other regions, I guess, because they're like more plastic in a way? Or is that not necessarily a fair assumption to make?
There's a really nice study of Baum et al., where they also look at structure-function coupling and how it changes during development. And yeah, there you also see that the default mode region plays a central role. At the same time, there's a lot of studies just showing a difference between unimodal and transmodal regions, but they do not then go into the nitty-gritty between paralimbic and heteromodal differences. So for example, like Daniel's, or like this gradient that goes from primary to default mode regions is also often compared to gradients going from unimodal to paralimbic regions. Just forgetting that actually the default is at the apex of the functional gradient, whereas paralimbic is agranular. So there is some divergence between these patterns, even though they highly correlate. So maybe that's, so, but yes, it's true that heteromodal regions, they change like longest also in development, and paralimbic regions are on the one hand hard to scan, making it also hard to interpret, right? But they're also like thought to be relatively plastic and variable. So yeah, it's tricky, but at least my take home from this study is to me that there are differences in the transmodal regions between these paralimbic and heteromodal circuits. So shall I tell now a little bit about meditation and what it does with your brain? It's already a bit late, but it's soon then over. So maybe some of you have some experience with mental training or meditation or mindfulness, or try to improve or change their mind with breathing techniques or other techniques. And yeah, of course, it is like a big interest in the field of neuroscience and also cognitive science to understand like what interventions can change our minds. And like in healthy individuals, it's often thought about like how to feel better, but also like in terms of therapy. So here, this was started as part of my PhD, but yeah, some of the work is still ongoing because it's such an interesting but also complex design. So you see that there were 2 training groups that did 3 times 3 months of mental training of 3 different exercises. So yellow is presence, and this is sort of thought of to stabilize the mind, and it's like this classical mindfulness that I guess many of you have done, like you breathe through your nose and you experience it. And this is thought to help with attention and like improve attention and interoceptive awareness. Then affective training relates to care and compassion, pro-social motivation, dealing with difficult emotions. And here you exercise like on your own, like opening your heart, so to say, to first people that you love, but then also people that you do not know really well, and then also people that you dislike, to understand that they were all, like, to say it in a bad way, they were all babies, you know, they all deserve to be loved. And that's kind of the idea that you have to like nurture. But also you need to speak about your emotions and listen to the emotions of others by having this dyadic exercise where you speak for 2 minutes about the negative things that you felt today. So I felt so angry at the German traffic when I cycled back home today, and I don't understand why all these cars are here, and I was so angry. And then something that made me happy that happened today. I think how lovely it is that we are all here, and like how it makes me happy to speak about my work and see what my group has done. And then you listen, you recognize maybe some of my emotions, and then it's, for example, Nadia's time to speak about her positive and negative experience, and I listen, and this should then nourish like compassion. And then there's perspective taking, and this relates to theory of mind, having the perspective on yourself and others, that we all have different minds and roles in our lives. So we spoke about me as a mother in the beginning already, but then it also quickly went to me in puberty, dancing, enjoying, and also drinking beer. And these are all different roles. So Malar is a father, for example, but he's also a professor, and maybe he's really into sports. So, and all these roles make him a little bit different. And this is like something that you need to acknowledge when for example, I react as an angry child when a paper is rejected. So then I'm not in the professional Sophie, but more as in the child Sophie. And so through talking to this experience and speaking from the inner voice of this experience, so for example, my paper got rejected and I speak from my inner child. So I was so angry. I yelled and I hit the door, blah, blah, blah. So, and then you recognize, this is the inner child Sophie speaking. And then you can tell a different story where you also speak of a different character in you. For example, the coach, or I don't know. So this is thought to help with perspective taking. And these were the exercises that our participants did. So we had 2 training cohorts of 80 individuals and also a retest control cohort that did nothing, but only got tested many, many times. And at the beginning of each training cohort, because I guess, well, my explanation now already takes 5 minutes, it's all rather complex. So for these individuals, we had a retreat of a whole weekend so that they could really get into these exercises. And they were all also quite committed. So they did this 5 times a week for 3 months and really were in the program. So the question for me was, how does this change our brain? So first, I could show that cortical thickness changes, so increases as a function of mental training. So you see that the yellow training, which was mindfulness attention, created cortical thickness changes in frontal regions, whereas the other training associated with affect created changes in opercular and sensory motor regions, and the perspective training had changes in the IFG, but also temporal regions. And controls, they showed, if at all, some decrease, showing already short-term aging as a function of 9 months. And also these changes related to changes in behavior. So I looked at within each training months, whether this related to changes in compassion for affect, attention for presence, anterior of mind for the perspective. And yeah, again, you see that this association with change in thickness with different regions. And it's interesting that you see the anterior insula for compassion, because this is also a region that has been functionally associated with affective skills, whereas the TPJ has been associated with these social cognitive skills. And yeah, presence and attention is a little bit more difficult to interpret. And the challenge, of course, in these longitudinal designs is also ceiling effects and baseline effects. But yeah, that was a bit harder to interpret, at least for me, but per se it's interesting that you see that these individual differences in skill improvement relate to brain change. So then the question became functional organization. So these are I'm now presenting the work that I've done in this study. So I've also looked at functional organization, first looked at the principal functional gradient. And what you see is that with affect training, regions in the ventral attention and dorsal attentional network, they increase, so they become closer to the default and frontal parietal network, whereas regions in the same network, they decrease following presence training. And you see that there's some insignificant changes also happening in perspective, but that was not what stood out. So for example, along this first gradient, this affective training and this mindfulness training were sort of their inverse of each other. Then I also combined the 3 gradients to look at more general patterns of integration and segregation of networks. And there I found that indeed you see that there's a difference between mindfulness and perspective taking this time, that mindfulness sort of segregated the regions, whereas perspective training integrated these regions, meaning that their functional connectivity became more similar to each other. And this also relates to bigger ideas about the global workspace and what is happening in dynamic brains when you either concentrate or relax. So this makes some sense. And then I also used a multivariate approach, linking the 3 gradients to behavioral change. And there you see that there also are associations with attention, compassion, and theory of mind, and that they to some extent overlap with task-based activations, meaning that maybe these functional organizational features in these 3 gradients also change as a function of training, as a function of individual differences in meaningful locations to some extent. So then this is maybe a secret, but I think it's recorded and it's fine. So I wondered a bit about these results, right? It's mental training and what does it mean? So in a last exploration that I did recently, I also looked at microstructural change. And what is to me, it's in preparation. I don't know if it will ever publish, but I wanted to share it with you anyway, because it helped my thinking a little bit, and maybe it helps you thinking too. So first, MP2 range is a great contrast to also probe microstructure in vivo and like related processes. But what is interesting is that you see these huge increases in presence following in microstructural organization. And you see it here also in the scatter plots. It's way more than controls, but also the other two modules. And this is particularly the case. So like this was a draft, so it's just my last slide. In heteromotor regions, whereas there was a decrease in the ideotypic regions. And you see that the inverse pattern takes place in perspective, where you see a huge increase in ideotypic regions, but a decrease in these heteromotor regions. And you see that in the middle is sort of affect, showing that along this cortical hierarchy, there's different changes happening as a function of training, as a function of cortical type. And this was actually sort of robust. So I saw this in both training cohorts, because I was rather skeptical about these effects, but you see that in both training cohorts, actually this microstructural map, which is T1Q, increases in presence and it decreases in perspective in heteromotor regions. So you see the same pattern emerging, which opens then the question like, A, what are you measuring? And B, like, how does this then relate to functional reorganization and these changes that happen in microstructure, and maybe that the environment may open up to our brains and cortical hierarchy. Yeah, just to sum up, multiple large-scale axes organize cortical brain structure and function. Genetic and phylogenetic factors interplay along these axes, and this has certain behavioral consequences, placing certain behaviors in certain locations in this two or like multi-dimensional space. And plasticity along this organization varies as a function of mental capacity trained, meaning that there may be hope for every one of us to learn social skills, or yeah, I don't know. I think it is rather intriguing, and like the more I look at different modalities, the more hopeful I also get in like what happens with the environment in the brain. So yeah, this is the end. Obviously, it continues. My lab is still growing and still developing and still moving beyond humans and macaques and more deeper also into microstructure and function of the brain. So obviously, this was not only my work, but also like the work of like a lot of other people that helped and supported me along the way. So this is like a somewhat older Zoom image of my lab, because a lot of it was, or everything was online until, well, it still is. And yeah, also a great bunch of collaborators, and also Max Planck funding my group in Max Planck, and Jülich funding my group in Jülich. And yeah, I'm also on Twitter, although a little bit less because I got a bit more busy. But like, if you're interested, then follow me on Twitter, and thank you for your attention.
Thank you very much. That was a fascinating talk and a lot of different results. So I guess would you have a couple minutes to answer a few trailing questions?
Mm-hmm.
Oh, awesome. So I guess if anybody's free to stay on, because Lonnie Kupo had a really great question. She was asking, do you have to control for the impact? So with the meditation study, do you have to control for the impact of the social aspects of the training? And did those undergoing more training build a community that the controls didn't receive?
That's an important point. So in principle, like the main analysis that I did was comparing training with training. So in that sense, you automatically control for the social aspects, assuming that people are similarly social, irrespective of presence versus affective training. And obviously there was some community building. You know, they went to lunch together, they hung out, they trained together. Many people, like on average, they trained like 4 or 5 times a week. So there were also a few outliers. But obviously these factors at the individual level may have a certain consequence to the brain, although it's at some level also hard to measure, which is why I try to compare also the different training cohorts, and compare only the main analysis that I do are like comparing differences between groups that actively train.
And then I had I had one last question pertaining back to the meditation training. And I was wondering if there's anybody who's doing something analogous looking at exercise and the benefits of exercise, like anybody in kinesiology who's pairing up with the neuroscientists maybe, or is that not current?
Yes, there are like people looking at like classical examples of juggling, or like also like what running does to your brain. There it's always challenging because you also dehydrate and this may also affect, you know, your brain. But yeah, like how, for example, exercise relates to cognition, or like how these bridges are built, or also how this like matters to your brain is like a whole branch of research. Also like thinking of the taxi driver study that started it all, right? So yeah, I guess we're just waiting of somebody like loading all Strava data of people that have UK Biobank and then correlating X with I. So yeah, I think there's a lot of that going on, and it's tricky though. Like I still use the data from my PhD, and like I wish I had my own data and my own design, but like it was so much work to first design this, and then the funding, and then like having all the teachers and doing it in the way it is. And I wish we had different contrast or a different setup. And it's, you know, it's always hard to get, well, you know, it's hard to get good data and good setups, and then even harder to set it up yourself. So yeah, it is, I think, an open quest also to get more longitudinal samples and understand how environmental variation may have an interplay with the brain.
And then we have another question by Breer Alexandra Rigby Dames. Great talk, thank you. I wanted to ask you if you're doing any research in brain transcriptomics as well as the genomics research. I just wanted to ask also if there will be access to the recording. Yeah, so the recording will be posted on YouTube, and I guess for the slides, I don't know if there's a way for this person to access the slides as well. So yeah. Sorry,
yeah, I guess they're in the recording, so I hope that this is sufficient. Yeah, about transcript, yeah, I do like also this 2D axis. I did relate this also to transcriptomic expression using Allen Human Brain Atlas. And so yeah, I'm also working with transcriptomic data and hope to also expand this a little bit further. Like, for example, with the asymmetry project, it was not possible to further investigate how this may relate to transcriptomic expression because there's only 2 donors that have left and right hemisphere. So that would make comparison rather tricky. But yeah, that took also, like, in terms of like valuable data, that it took like ages for them to collect these 6 donors, right? So, and like, I don't want to be a data leech, but like I use a lot of open data at the moment, and like these more in-depth profiling that takes a lot of time and money to collect and use. So I'm also now running my own study slowly to also give something back to the community. And yeah, there is like also brain span. There's a lot of resources there, although yeah, obviously it is still tricky to bridge also different levels and understand what's going on, knowing all the individual differences also.
So I guess we're a bit over time, but there was one, so I guess anybody who, yeah, there's one last question, but I guess we're a bit over time. So maybe it could be, but yeah, taken another time, but so yeah, thank you very much. That was a really wonderful talk.